collaborative project I've been doing with some microbiologists at LSU Health Sciences. And this is centered around looking at microbial communities. So the background on this work, we've all heard a lot about genomics and looking at the human genome and whatnot. That really doesn't tell the whole story. So there's genomics, proteomics, transcriptomics, and interactomics, and all of these things tend to focus on the whole picture of a single organism, like the human. But they're actually organisms like the human live in communities and they interact with other things in that community like bacteria. And in order to study such a community, what we want to do is look across the genomes of all the communities or all the organisms in that community. So this is where metagenomics comes in, and metagenomics is essentially the study of metagenomes, or all genetic material across all organisms sampled directly from the environment as opposed to being cultured in the lab. And this allows us to do a lot of different things. One, it allows us to study the communities in their natural environment, and it bypasses the need for lab cultivation. A lot of the things that we want to look at can't really be cultured well in the lab. So an example of one of the environments you would look into is the human microbiome. And the human body is heavily colonized by bacteria. So there's bacteria that lives on the body, on the skin, within the gastrointestinal tract, urogenital, airways. And this figure is just taken from NIH Human Microbiome Project, which is a large project that is funded by NIH to study and characterize the human microbiome. And to give you an idea of the magnitude of this problem, there are roughly 10 trillion bacterial cells that live on and within a human body versus 1 trillion human cells that make up the body itself. So one of the things that they looked at, the skin microbiome, they sampled various locations across the body. And what each of these pie charts represents is the bacterial community that was found on a given site. Like, for instance, this is the bacterial community they found on the heel of the foot. And you can see that's well dominated by Staphylococca. But that sort of community is very different from the sort of community that lives on the face, for instance. So how do we study these communities? In effect, we look at this single molecule, the 16S ribosomal RNA, and it's a gene that codes for the ribosome. It's a highly conserved gene because the ribosome is essential. And so there are actually what are called universal PCR primers, which can be designed, and they will amplify up the 16S ribosomal RNA gene from any bacteria. They're called universal. They're not totally universal. There are a few bacteria that could fall through the cracks. But largely, you put in these primers, you do your PCR, and you amplify the 16S ribosomal RNA gene from all these organisms. And then, of course, you're going to sequence that with high-throughput sequencing technology. So the basic protocol for these experiments that we do, the samples are collected and prepared, and you're typically going to have many samples. What we're looking at primarily is diseased versus healthy patients in a variety of different diseases. And then you use your universal primers to amplify the 16S ribosomal RNA gene from your community. Then you use next generation sequencing of that amplified DNA. And of course, those reads are going to begin with the highly conserved region that the universal primers were designed to. But the good thing about this is that we need to use high throughput sequencing technology that gives us long reads like 454, because we can read into what are called hypervariable regions, which have mutated more along the line of evolution. So these hypervariable regions actually contain the phylogenetic information that we're going to need to speciate these sequences. And essentially, the sequence of the hypervariable region for a given read is kind of like a calling card for that organism, and it's matched to a database to say, okay, this particular sequence came from Staphylococcus. So there are a number of caveats with doing this. One is that the 16S ribosomal RNA gene copy number is not uniform across bacterial species. So if you were to use that as an estimate of relative abundance between different bacteria, you could really fool yourselves. But So what we end up doing is we do what we call semi-quantitative phylogenetic community profiles. So we can compare the relative abundance of a given bacteria across two samples, let's say a diseased patient versus a healthy patient. And if we see 5 times as many sequences of that given bacteria, then we can safely assume there are roughly 5 times as much of that bacteria in the diseased patient versus the healthy. Of course, once it's been normalized to the total number of sequences. And then we go on to analyze it, and this is where I come in. So the biological researchers I work with have gotten up to this point. They've done some sequencing, and we take those sequences of the 16S rRNA reads and we assign them to their origin. And for this right now, what we're using is a Ribosomal Database Project. They have an extensive database of 16S sequences from all different bacterial species. And if I have time at the end, I'm going to talk to you about a possible alternative to this. So the process is really complicated by a variety of factors. Of course, there's some significant error rate with pyrosequencing, anywhere around 1%. And 454 sequencing in particular is susceptible to what are called homopolymer errors. So that makes it a little bit difficult to differentiate these sequences all the way down to the species level. There's also the issue that annotation is sometimes poor for a class or a variety of sequences, so we may not really know the details of what some of the sequences we're pulling out really are. And lastly, one thing that's really big with the biologists I work with is that often the top hit or the best hit that you get when you try to match the sequence may not be the right hit. It may come out of the database and tell you that something you took from the human gut looks like an acid mine drainage bacteria, whereas the next closest sequence is something you might expect to find in the human gut. So all of these things present challenges, but I'm going to motivate what I'm actually going to talk about with an example of what we could do with the software that we're building. So one of the experiments we were dealing with is a disease called bacterial vaginosis, and women will come in, they have bacterial vaginosis. You take a sample of the bacteria living in the vagina, and you want to look at what's present in that community. So there are a couple of different methods you could do that. One that they used a cotton swab to get some of the bacteria, and then they sequenced it. And another one they used an actual scrape to take tissue. And of course the scrape is going to be more invasive, more painful than the swab. So one question you'd like to ask, can we tell the same thing from the less invasive swab as we see from the scrape? So I've just got some examples here. These were 4 different patients, sample 1, sample 3, 6, and 7. And on the top we have the community that we found via our software with the vaginal scrape versus the community we found with the vaginal swab. And if you look at these patients, you can see that the distribution of bacteria that we're finding in a given patient with either technique is relatively similar. So we could conclude that perhaps we can go to just using the swab. Another thing that we'd like to know, well, sequencing is getting cheaper and cheaper, but it's still not a trivial expense in your biological experiments. So the question is, how deeply do we need to sequence an environment to be confident that we've got a good distribution of the bacteria that are present there? And so we can generate these what are called rarefaction curves, and on the x-axis we basically have the number of sequences that we've done. So we've sequenced 5,000 reads versus 10,000 reads. And on the y-axis we have the number of different species or families or orders, classes, or phylums that we've found. So getting more specific as we go up. And what we want to see from one of these curves is that it's kind of plateaued by the time that we stop our sequencing. If we actually saw a curve and it was still rising significantly, if we'd only sequenced to this point, we know that we need to go ahead and pay for some more sequencing or do some more samples to get a better look at the environment. So what we're doing is we're developing this framework, this software framework for analysis of microbial community composition. And we had a lot of goals for it. One was we're working with biologists. They don't want to have to learn a lot of messy tools or use a lot of command line features. So we wanted it to be pretty automated. The standard analyses that we do, like rarefaction curves and community composition pie charts, are all initiated upon load of a data set. We wanted to be very easy to use, so we built a web-based interface through a web browser. Our server can actually be accessed just by Internet Explorer, Firefox, whatever the user wants. and the actual framework is running on a server machine. We wanted to be efficient, so we wanted to be able to process millions of sequences very quickly, and we know that this is going to grow and grow as sequencing becomes cheaper and cheaper. We also wanted to be very extensible, so if there are new methods, we wanted it to be easy to be able to integrate them into our framework. And we want it to be graphical, because a lot of the times what the biologist wants, they do their experiment, they upload their data, they want to see what does it look like. So it spawns off these pie charts and publication quality figures right off the bat. And lastly, we provided a lot of interaction so they can do collapsing and expanding of figures and parameters and toggles. So this is a general overview of the system. Everything starts up at the top with the researcher providing their own FASTA file of 16S ribosomal RNA sequence reads. And then we perform the classification currently via RDP. And then at that point, all these standard analyses are kicked off. They get pie chart representations of their communities. They get information about their individual samples. They get actual textual-based information that they can hover over and see what was there. And then the researcher can interact further. So let's say they have some sort of hypothesis, and we're dealing with some disease. Maybe we want to look at whether weight is a factor. So the researcher can generate these what are called sample maps, and taking all their samples, they can color code them. Let's say maybe the black is overweight and the white is normal. And then the framework can kick off some more analyses to generate heat maps and see if indeed there is a significant separation between those two factors. So this leads us to the Ribosomal Database Project. It's currently what we're using to classify these sequences, and it provides us with classification at all the different levels of the taxonomic hierarchy. Of course, the kingdom level wouldn't be very useful to us. Everything we'd be looking at is in the animal kingdom, so we don't bother with that. But we get our classifications at phylum, class, order, family, and genus level. And as I said, it's a little bit hard to get down to the species level due to some of the sequencing constraints. So some of the future work that we're working on right now is another approach other than using this ribosomal database, where we're looking at a pure sequence-based front end, and I'll motivate that at the end of the talk. So some other things that we show in this framework, for each sample, each of these on the x-axis is a sample, we show the total number of sequences so that the researcher can easily see whether they didn't get enough sequences from a sample to make any valid conclusions, or if they need to sequence further a given sample. Diversity indexes are computed, so these tell us how diverse is a given community. If everything in the community was Staphylococcus, it would be very low diversity. If there was a lot of different things in the community, it would be very high diversity. Then we generate all those rarefaction curves I talked about. Of course, it's not very useful to view them all together, so we allow the researcher to go and grab individual rarefaction curves for each sample. And then they have, of course, the pie charts. And the pie charts go, again, all the way from genus, family, order, class, phylum. And what you can see is in each column, we have a different sample. You can see, as you get more and more specific with your classifications, how these different groups break out into different species or phylums as you go down from phylum to genus. And here's just an enlargement of what that looks like. So you can see at the very abstract level of phylum, we don't have a whole lot there. We move to class level, things don't differentiate much. We move to order level, again, things don't differentiate much. But once we go from order to family, we see this large order here breaks up into different families, and the families break up into a lot of different genus. And of course, we don't yet have the species level. And some of the future work is to develop a more smart coloring algorithm to have uniform coloring across taxonomy levels, because of course this class here is what led to that order there, but they're different colors. And that just gets into developing an algorithm to uniformly color things. And then the researcher can generate those sample maps to investigate those hypotheses that they want, so they can enter in any information and they can just say, I want to look at these samples versus those samples. But that requires a lot of interaction, so some more future work that we want to do is to allow an automated importation of some metadata to provide easier creation of these sample maps, and also possibly automated hypothesis testing. And then once you've generated or entered in your sample maps, in this case maybe the red is a diseased patient and the green is a healthy patient, you actually get heat maps of all of the bacteria that are in a community and ordered phylogenetically on the top to see whether we have a significant differentiation between your color codings. And you can view this in another way. You can view it with the actual pie charts put on a dendrogram, and some of this got cut off a little bit, but this is actually a dendrogram saying that these two samples were most similar than these two, and so on. And so you can see whether diseased or healthy patients are grouping together. You also get the diversity indices. You can look at those on your sample maps. So if we have said we are looking at obese patients versus normal patients, you can see whether there's a significant differentiation in the diversity of the communities between those two groups. And we get dot plots. So we can look at individual, for this case, genus, and see whether obese versus normal is significantly different for each individual genus. The architectural overview of the framework, we have a dedicated server that is on one side of things, and that has the databasing. It has a core framework of all the functions. It has all the data processing modules about sequencing the data and things of that nature. And then we have our API, and on the other side we have this nice web client we developed. We have a command line client, and it's flexible enough that we could develop other clients if we need them. And so some of the things that we're going to be doing to enhance this framework I already talked about. So this leads me to my last question, which is, are we really hamstringing ourselves by staying entirely with taxonomy? So the first step when we take these sequence reads is we use the RDP database to assign a taxonomy to each read. And that leads to a very coarse assessment of diversity. Of course, coarse should be C-O-A-R-S-E there. And the 16S is really just used as a marker or a footprint that that bacterial species was present. But taxonomic classification, it turns out, can be a large oversimplification, which is going to extract away important details that we may like to look further into. And so the question is, maybe we could ascertain a little bit more information from differences in the 16S sequences than just putting our blinders on right at the front with a taxonomic classification. So I'll just use a small case study, the role of the Gardnerella in Gardnerella vaginalis in BV patients. And so Gardnerella vaginalis was thought to be the causative agent initially of bacterial vaginosis. And these are bacterial vaginosis patients on top. These are normal patients on the bottom. And what you can see, Gardnerella vaginosis is in the red here. It's a large component, or a larger component, of women that have the disease than those that don't. But if you look more closely at women that don't have the disease, one of the things you see is that Gardnerella is still the second most abundant bacteria present. So the question then becomes, why does Gardnerella not cause problems in healthy women? And are all Gardnerella the same, or are we talking about two different things? Well, there was a paper late last year, that talked about this whole issue. And what they showed in this paper is that there are two different types of Gardnerella. One that is cohesive and can form biofilms, and another that's dispersive. And the cohesive type was present in all BV women and their partners, their sexual partners, but it was also present in 13% of women hospitalized for other reasons in BV. So there's strong evidence that the cohesive type of Gardnerella is actually the bad causative agent. So what can the 16S sequence tell us? Well, the difference between the cohesive and dispersive types of Gardnerella would be lost if we just go simple taxonomic classification like we've been doing. It would just say these sequences are all Gardnerella, and you wouldn't see anything else. But if we use a pure sequence-based approach where we look more closely at the sequence, can we discern such differences? That is there a difference between the Gardnerella sequences from healthy patients versus BV patients, even at the 16S ribosomal RNA level? So one of the things we did to just take a real quick look at this was to do sequence-based clustering on the 16S ribosomal RNA sequences, only the ones that are from Gardnerella. And we had 373 such sequences from 13 healthy women, And what we've done on this chart is the outermost tier is actually 100% identity. So you can see that there are some individual sequences. The size of the dot is the number of sequences in that cluster, and you see some small, larger dots there representing that we had multiple instances of individual sequences. Then we go into the next tier. We've grouped everything into a cluster if it's at least 98% identical. So what you see here is you see that almost all the sequences group into a single cluster within the healthy patients, the Gardnerella sequences. And then, of course, that cluster maintains itself going from 98% identity to 96 to 94 to 92. And just for your reference, 97% identity is about where the cutoff point is for bacterial species to say that two bacterial species are different. So we're looking at, if we went with RDP, things would be classified in between this 98 and 96 tier. So what happens when we look at diseased patients? Well, of course, we had more diseased patients enrolled in the study. When you do a study, you get people with a disease. It's harder to get normal patients. But we had 39 BV patients, so we took a random sampling of 373 of those sequences, and we did the same clustering. We saw something far different. At the 98% identity, we actually got two significant clusters. And then at 96%, they merged. So we thought, well, maybe we're just fooling ourselves. Let's try this again. Maybe the 373 that we picked were just luckily going into these two different groups. So we did it again. We saw almost the same exact thing. Two different groups at the 98% level. We did it yet again, and we kept seeing the same thing. So we thought this is a pretty good indication that maybe there are different types of Gardnerella present, and maybe we're looking at one group being cohesive and one being dispersive. But this is a very coarse view of things, so we wanted to look a little bit more closely. So we could identify portions of the Gardnerella 16S alignments that were information rich, meaning they had different nucleotides in a lot of the sequences. And if we did that, what we could do for each patient is we could form what's called an oligotype, talking about the different nucleotides that were present at each of those information-rich positions. So we had three patients we looked at, and what you can see is that each of these has a significant oligotype up at the top, and they have different versions of Gardnerella there. But this study included both male and female partners, and the most interesting thing we saw, because there is a little bit of a theory, a female patient would come in, she'd be treated with antibiotics, the BV would go away, she'd go back, and she'd, come back in with BV. So there was a theory that maybe the bacteria is being passed back and forth to the sexual partners. So if we looked at the male partner that matched with each female, we saw that the same oligotype dominated their Gardnerella sequences. And we saw this throughout all the patients that we looked at. So that provided strong evidence for us to continue looking at this, and we're still looking at that now. So the conclusion is that if we develop a more sequence-based approach to identifying these sequences, instead of going through RDP, maybe we can get some more information out of the data than we're getting at present. And just to wrap up, I'd like to acknowledge my graduate student, Murat Arin, at University of New Orleans. He's done the primary part of this work, developing the framework, and my biological collaborator, Mike Ferris, in his lab at LSU Health Sciences. And there are some of the references. Thank you. Right. So the first question was, why do we think the 16S ribosomal RNA gene is relevant to these diseases? And actually, we don't. So the 16S ribosomal RNA is just something that we use as a calling card to determine which bacterial species are present. It's not actually that gene that has any effect on these diseases. We're just using it to find out what is in our community. So it's just kind of a calling card. And the second question was, what was the second question? Oh, right. Right. So the second question is, well, why don't we look at the directly related genes that might be factor in these diseases? And that is, of course, the Holy Grail. So right now we're just doing 16S ribosomal sequences to look at communities. But if you wanted to spend a lot more money on your sequencing, why not just sequence the whole genomes of all the organisms instead of just pulling out the 16S? And there are people that are doing that, but we're not quite yet that well funded. Metagenomics. So what he's suggesting. Of all the bacteria. So they're taking all the genetic material and they're sequencing all of it. As opposed. Oh, for us, so we're taking that whole community and we're amplifying only the 16S to figure out how many different bacteria are there. But depending on what you're looking at, bacterial vaginosis, you'll see maybe 6 categories, 7 categories of species. But another disease we're looking at, necrotizing enterocolitis in infants, you actually see a lot more diversity. And there, the diversity is actually a factor that is negative for the disease. So the babies need to develop a very diverse flora in order to hold off that disease. And it's babies that have very few things in their community that actually fall prey to necrotizing enterocolitis. Well, you mean the technology we built it on, or where are we housing it?
Technology. I know it's turbographs, pylons.
Yeah. So we're using Python, and we're using some of the libraries for Python, and in particular, we're using Django to produce a lot of the web-based interfaces. My graduate student loves Python, and it worked out pretty well. So as long as things are working, intervene and say, no, you need to take another course.
Your graduate student has good choice. Yes. Okay, let's thank Chris again. Okay. Dan, our next speaker, we have a tag team.